this from the outside comes in, because what they don't show you in this picture is that there is a cell. There are cells here, so the cells would diffuse. Carbon dioxide would diffuse out of the cells and then move into the blood. Waste that are also produced by the cells will move and then move into the blood. Excess water that was pushed out will also move back in, and then these would fuse with other with other capillaries to form venules. This is where it starts bringing things back to the heart. So veins normally carry blood towards the heart. Arteries carry blood away from the heart. Then I showed this picture. Then if talking about the heart, we say it's something as an organ that is about the size of our fist. It has its own blood vessels that feed the muscle itself when you cut it open. It has 4 quadrants, two top quadrants here and lower quadrants. Top one's atria, one atrium, lower ventricles, and looking like this. This is actually the left side, and this is the right side. Then we have here connective tissue that are holding. The top chambers are connected to the lower chambers by valves. A valve is basically something that controls one-way movement of things and then prevents the stuff from going out the other way. So when blood comes here from the atria and it moves to the ventricles, the lower versions, when the heart is contracting, when it contracts, from the ventricles here, we have the blood connection to the blood vessels leading out of the heart. This one here is the aorta, which is the large artery, this is another artery here, and this aorta here is painted red, meaning that it is carrying blood that is rich in oxygen. This is also an aorta, but because it is painted blue, because it is carrying blood that is rich in carbon dioxide. This is the blood that came from the rest of our body and needs to be sent to the lungs for oxygenation. So blood comes in to these top quadrants, fills in, and then goes below here in the lower ventricles. And then when the heart contracts, it squeezes stuff off the blood. And because of these valves, blood cannot go back into the ventricles. The only way it has to go now is to go through these blood vessels, the aorta and the pulmonary artery that is leaving the heart. So again, know the names of these chambers. This is a better structure, better view of it. And these connective tissue, sometimes they will call this atrioventricular valves. Atrioventricular valves. This one here has two sets of connective. You hear they call it, sometimes they'll call it bicuspid valve. This one they'll sometimes call it tricuspid valve because of these connective tissue that is holding them together. And the thing to also note is that the left side of the heart, which is like this side, is separated from the right side. So blood can never cross from these chambers into these other chambers. And the separating muscle that separates the left from the right one is called the septum. And also, depending on if you look at how the structure of this organ matches its function, the walls of the right atrium here, this is like muscle cells. The walls here are less thick. The muscle is not as thick as the one you find surrounding the left ventricle. Why? Because the blood that is in the left ventricle, when the heart contracts, this blood here moves and goes to the rest of the body through this aorta. Meanwhile, the blood in the right ventricle has to go just not too far, just goes to the lungs, which is not too far away from the heart. So there's no need for the contractive force to be there's no require for too much force to be on the blood, since it has to go only a short distance. This is a better separation, showing that this one here, the left ventricle, the blood, it doesn't show the connection here, but there is a connection that leaves and connects to this aorta. This one here shows here that from here, when it contracts, the blood will go this direction. Know how to label and all of this and explain it. So this, I said, this is a good diagram to remember and be able to understand how the movement of the blood from the right side, it moves to the lungs and then back to the heart. Things that have to do with the lungs, they call pulmonary. Things that have to, as it's coming back, so pulmonary artery. This is the only artery that carries blood that is poor in oxygen, carrying deoxygenated blood. That is the only artery. Ordinarily, all arteries carry blood that is rich in oxygen. And conversely, this is the only vein after oxygenation of the blood in the lungs, the blood is coming back to the heart. This is the only vein, the pulmonary vein, that carries blood that is rich in oxygen, oxygenated blood. Ordinarily, all the other veins here, bringing blood back to the heart, carry blood that is poor in oxygen. So you have the aorta, blood leaves the aorta, some of it goes to our head and arms region, then the rest of it goes down to our intestine, to our legs, and all of that. Then again, after at the capillary level, they start fusing back, and then they will form these veins that bring blood back. This going from heart, lungs, back to heart, pulmonary circuit. From heart, the rest of the body, back to the heart, systemic circuit. Again, it talks about all of this. Now, we talked about visualizing blood flow. There are videos that will allow you to be able to do that. This video, the link should be active for you to see. How structure of vessels and heart match their function. We briefly just talked about that. The thickness of the walls and the thickness of the walls of the blood vessels, and looked at the thickness of the walls of the heart, the left and the right side. Now, in terms of heartbeats, our heartbeats like so you hear when the heart contracts, they call that the systolic phase. When it is contracting, the ventricles squeeze the blood to move out. And then that contraction is followed by a relaxation called the diastolic phase. The chambers relax, blood moves from the top aorta, from the top atria, and come down to the ventricles. I said this circle of contraction and relaxation happens about 70 times a minute. So what is it that accounts for heartbeat? We said there is a node called the sinoatrial node, and then there is another node called the atrioventricular node, this one, and then you have this nerve fibers called Purkinje fibers that swam the rest of it, they span the almost like from the middle to the lower portions of the heart. So these cells here would produce an electrical signal that would now go and stimulate this atrioventricular nerve, and then it itself will take that again, push out some other electrical signals which will be carried by the fibers and the Purkinje fibers, and it would spread out to all the tissue, the muscle tissue here. And these electric call action potentials, they would make these electrical signals will make the muscles to contract to all together at the same time. We said this contraction can also be controlled or influenced by some chemicals. Like if something freaks you out, if you get scared sometimes, and then your heart starts beating fast, because some chemicals, I said, like epinephrine and norepinephrine are also controlled. When these chemicals are present at elevated levels in the blood, they can also make the blood the heart to beat faster. And this beating of the heart can be captured by an electrocardiogram. And if we understand that we have a contraction phase, and then we have a relaxation phase, you would see that on the electrocardiogram, you could sense when the heart is contracting, you could sense when it is relaxing. And then if they measure your heart and they can tell if something is not happening right in terms of the electrocardiogram, the graphical image that is produced by this. Based on that, they could tell, oh, there might be an issue in your heart in this area. So they use this EKGs, they call them for diagnosis of. And then we also measure blood pressure to also inform how the heart is beating. Sometimes they use these instruments. They will tie some on your hand, they will tie some on your wrist, and then they would look at when the when the heart is contracting, what does the pressure read? Blood pressure again is the force in which the blood is pushing on the inside parts of our blood vessel. So when the heart contracts, the blood is squeezed out. As the blood is squeezing out, it hits the walls. If you have those collapsible power water hoses, when you turn the water hose, as the water rushes out of the faucet, it goes into those pipes and stretches them out and makes it stretch out. It's similar to how when the heart contracts, it pushes the blood to be able to move. And so what the blood pressure is basically measuring is the force on which this blood pushes on the inside walls of the blood vessels. And they would measure that when the heart is contracting and when the heart is relaxing. The average during contraction, the average blood pressure reading will be 120. And then the one when the heart is relaxing, the diastolic one, is about 80. For adults, it will be slightly different for babies, and also sometimes. So when they say this is average, because it might be about 120 something for other individuals, or slightly lower, and for the systolic, and slightly higher and slightly lower for some individuals. However, if it goes beyond certain parameters, it can be an issue. And some of the things that could cause blood pressure to be high could be a blood is because it might be so thick that the heart would need to push more, trying to put more force for it to move out. Or some of the other things could be is that the openings inside their blood vessels might be slightly narrow, so that when you have a very narrow tube and you're applying pressure, the same amount of pressure, then the pressure of the force that the liquid is exerting on the inside part of the blood vessel is hard. So for example, if you open your water hose again, the water is flowing out normally, you take your finger and you close you slightly close the out, one of the opening of the water hose, the water would eject out at a higher pressure. So if you have a narrowing of your blood vessels, it can cause increased high blood pressure, high blood pressure readings. This is average, normal. When it goes around, I said for the systolic, I said it could be slightly lower, but when it starts getting to 95, it would see a hypotension, and then if it goes above 110, now it starts becoming to 139. It starts like you start having hypertension, and this becomes issues where you need a medical intervention. The blood pressure I was saying here again is lowest here and highest here. Blood coming back to the heart is thanks to skeletal muscles squeezing on the blood vessels. Veins have valves, one-way valves, preventing backflow. When the muscle contracts, it squeezes the veins, and then the blood will move up this section. When the skeletal muscles relax, the tendency would be for the blood that was in the veins to come back. But since they have valves here, it cannot come back drain backwards. So this continuous contraction of the skeletal muscles help to bring blood back to the heart. I said you have circulation, the heart helping in pumping blood throughout the rest of the body and to the lungs, but the heart itself too also needs to the muscles, the cells that make up the cardiac myocytes, or the cardiac cells, to also need blood to be able to give them the nutrients and the oxygen it needs and to get the rest of the waste. So you have coronary arteries and coronary veins that feed the heart muscle itself. So the hepatic portal system, it has to do with our liver. When a connection between a blood vessel that feeds our liver, said the system that brings blood from the digestive system that is rich in all these nutrients to the liver. Then the liver would use that and take the proteins and store some of the glucose as glycogen, and then breaks down some of the red blood cells that are too old. And so it does all of these things. So we have a connection, a hepatic portal system that connects to the liver. Some disorders of blood vessels, I put them here. We've talked about them: hypertension, atherosclerosis, stroke, heart attack, or aneurysms. You can go through and look at what all of these are here. Ballooning. We went through all of this the last time. And then, did we go this far last time?
I don't think we went here.
Okay, so then let me take a little. So if I believe we ended somewhere here. If you look at disorders so if we have blood vessels, we have the heart. So let's look at these disorders that would happen in some of these structures. Talked about hypertension. So I said hypertension is called high blood pressure that results when blood moves through the vessels at a rate that is higher than normal. And some of it could be caused by arterial plaque, arterial deposits of this cholesterol, saturated fatty material that blocks the inside of the arteries. You can liken it to having stuff blocking your pipes in your house and making the water not to come out properly in large volumes, but coming right in higher pressure. So we say that if somebody's blood pressure keeps being above 140 and over 90, is considered hypertension. And when you have this continuously, you could have a heart attack as a result of this. Or you can even have like some of your blood vessels breaking or open. Because if you remember that we said that at the capillary level, it's only one layer, one cell thick, the walls of the arteries. So if the blood pressure is too high, it can easily burst them open. And when it bursts them open, it's a big deal. Because what it would mean is that the blood vessel that was feeding that particular group of cells, that tissue, will not have a supply of nutrients and will not have a way to pick up the waste that has been produced by that area. So you have that tissue in that area will die. This is a normal artery opening. The lumen is clear, it's all rounded inside, but look at what will happen if plaque builds up. If plaque builds up and blocks a big portion of it, then out of a sudden now, blood has only this small path to be able to come out through. So it will make the pressure to be high. And then sometimes, since this plaque is not something that is attached, stuck, or part of the blood vessel itself, sometimes the blood could make this plaque here to break and start flowing, moving in the blood vessel. It's almost like you have a small ball rolling in a pipe, and if the pipe is larger, the ball keeps moving. But out of a sudden, if it meets an area where the pipes are smaller, it will just like the plaque is enough to totally seal off the blood from continuing in that small vessel. Again, the net result is deprivation of the tissue downstream from having access to blood. And then when you have that happen, you have like a heart attack, or a heart attack would normally involves like when you have if that happens, a piece of tissue, part of your heart is dying, and the heart normally works by, it's almost like every cell in the heart contracting together and relaxing together, contracting together. But if a few of these cells are dead, then that idea of contracting together cannot happen very well because of the dead piece of section of the heart. Then you could have a heart attack happening there. Or if it happens in the brain, where you could have a situation where some of these blood vessels rupture in the brain, you could have a stroke. Again, because if that part of the brain that was damaged, the nerves that are supposed to be living from there to control certain parts of your body will not be able to function, and then they will die, and then you have some of the symptoms that result from a stroke. A stroke is also known as a cerebrovascular accident, or usually occurs when a cranial artery is blocked or bursts. Part of the brain dies due to lack of oxygen. And we said, since the brain is controlling the parts of our body, unlike muscles, on what to do, like for example, people who have this might have numbness of the hand because the nerves, they are messed up, of the face, and difficulty speaking because the nerve cells that are supposed to be sending signals to control our face, to control our hands and speaking, all those are messed up, so that system doesn't work very well. And then also, I said heart attack, also called myocardial infarction, or this just myo is the muscle, cardiac muscle. Part of the heart dies due to lack of oxygen. So you see here plaque. It starts building up, making the blood, the red blood cells, you can see them here, probably having challenges moving, can begin with an angina pectoris, which is pain that radiates down the left arm due to the blockage of the coronary artery. Aneurysm is a ballooning of the blood vessel. So you have atherosclerosis, and hypertension can cause weakening of the vessel due to too much pressure on it, and then it will balloon. And then I said the most commonly affected is the abdominal part, or the part, the vessels leading to the brain. So when you have all of these issues with blood vessels, how do they try to resolve them? There is sometimes when you have all of these clots in your blood, or they would give you some blood thinners, they say something that will go and try to break some of these clots. There's one called tissue plasminogen activator, TPA. I think it's made by a local company. I think it's Genentech that makes it. So they give you this, it goes and try to dissolve some of these clots that are existing in the vessel. Or if the vessel is actually blocked too much, they would say, so imagine here that this was plaque here that is blocking this vessel. Sometimes they would do, say, a bypass surgery. So they will take a vein from your leg and connect it from the part that is before the blockage. Then they would connect it to connect the part that was downstream from the blockage, to be able to make sure that it still has vessel. It's almost like a bypass, right? Instead of going directly the original vessel, that original vessel is blocked. So they will take another piece of vessel, blood vessel from our leg, and connect it, sew it, and move the blood to pass and go back to the area which it was supposed to be connected to. And then sometimes they will take like a stent, which is basically a wire mesh cylinder that is threaded into the blood vessel. When it gets to the clogged area like here, and then they would make the balloon to open. They would make it open, because this here is plaque here is based on fatty material, cholesterol. So when they go there, they could any pressure that is on there could dislodge, kind of break it into smaller pieces to move along. So you can see here, I said a stent with a balloon. I said build up of cholesterol, partially blocking this vessel. And then here they put a stent here in like a small wire that goes here. And then when they look at it under the microscope, as soon as it gets to this area, and then they will inflate the balloon. That means it's almost like they pass air, and then the balloon would increase. When it inflates, now it squishes on the plaque. And then when they relax it, the smaller pieces of fat now are able to move through. But now this stent, now that is almost like a balloon that is made of some tougher material on the outside, that when you inflate it, it breaks, squeezes these things open, and then the balloon kind of sticks. Because of the tough material, it kind of sticks and kind of keeps it open. And then the blood continues to flow. They call this balloon angioplasty. And then sometimes the heart gets so messed up that they could actually find, let's say, disorders that heart failure when the heart is no longer working. Sometimes they will have a replacement heart, or sometimes you would have situations where even we said there were some veins, there were some the atrioventricular valves. Sometimes the valves are not working properly. They will have to change them. And then sometimes now you have a pacemaker that if your heart is not sending those signals to make the heart to contract, then you have a pacemaker that will be sending out those electrical stimulations for you. But the general speaking is that there's been a lot that is known today on how to rescue people. People don't just die from heart attack quickly. They have ways that they can do to resolve their condition, but it's a very involved and very complicated stuff. But so it's not an easy thing to resolve. So I said cardiovascular disease and prevention. There are certain things that we know that have been associated with people have diseases that are involving the cardiovascular system. We know that if you smoke today, I don't care what you smoke, if you smoke anything and bring anything other than clean air into your lungs, you could have issues that would affect your cardiovascular health. Alcohol intake. If you're listening to the new reports that they were just talking about not too long ago, I think a couple of weeks ago, they were talking about alcohol. They used to use the word alcohol abuse, saying that one drink a day is fine and all of that. Abuse meaning if you're drinking a certain number of drinks a day that is higher than the one drink a day or something like that. But now they are even saying that the use of alcohol has been correlated to not only to cardiovascular health, but now they're talking about cancer health. So alcohol intake, we know, can affect your cardiovascular system. But also, we also know now that alcohol in any amount there's a relationship the correlation that I've been seeing now to what increase in cancer in those individuals that use it. Sedentary lifestyle and being obese, overweight. When you are obese and when you sit down too much, then they remember that we said walking around helps the skeletal muscles to contract to be able to make the blood to move. If you sit down too much, your skeletal muscles cannot be function properly for the blood to come back to the heart. That becomes a problem. Poor diet. Depending on the things you eat, if you eat a lot of saturated fats and cholesterol containing material, it can go and cause that blockage for you. Stress can also cause it. Poor hygiene can also cause it because some of the bacteria can go there and pass and get into your blood vessels and go and cause issues. And if you think about, yeah, these things are most of them are lifestyle issues. Whether you smoke is lifestyle. Do drugs and alcohol abuse is lifestyle. Whether you do activity is lifestyle. Poor diet, if you are feeding only from McDonald's and eating French fries and all of it, is lifestyle. Stress sometimes is difficult to avoid. Dental hygiene, if you don't brush your teeth, again, is something that we could control. But it almost looks like these are personal issues that people have to deal with. But if you think about why would it be beneficial, or if somebody does all of these things, is it anybody's business or is just their own business? Should we care about whether other people, fellow members or neighbors, are healthy? Is it something that we should worry about as a society or not? This is a question I'm asking. Is it anybody's business if people are not having a good time?
I would say yes, because sometimes people don't always have the same opportunities for having a healthy lifestyle. So it is other people's business to like make sure that the communities can help each other.
That's for being nice. That's great. Yeah. But if I choose to smoke and drink all I want, and I said, don't bother me, is because this is a free country in quotes, and you say that is that should we leave them alone or does it affect us either way? Any other person? It's true. You can give advice to people and they don't take it, and that is their business. Is it totally, if they're only their business, if we give them advice and they don't take it, does it affect us in any way though? Okay. Any other thoughts? Would it affect us in any way? I would think that the only way it would affect us is if you think of the way we pay for the way we pay for medical care is that we use insurance companies to mediate, to interact with our healthcare providers. And the cost of insurance is a function of how healthy the people as a pool that are being insured are. If you have many people who are sick in your society and the insurer wants to provide insurance for those, the insurer will consider that it looks like many of these people are going to be sick, so they're going to be using a lot of resources from medical care providers. So the insurance premiums are going to go up. And when they go up, they're going to go up for everybody. On the other hand, if you have people who are more healthy in the insurance pool, they say, something, we still make money here because many people will not be going to the hospital or to the emergency room because they are sick. In that way, we will not be paying out too much. So we don't need to charge too much premiums. So if you look at that from that vantage point, it's almost like it's cheaper for us to encourage our neighbors to be healthy, not just because it's a nice thing for us to do to make sure everybody is well, but also for almost like a personal reason in the sense that I don't want my insurance rates to go up. But needless to say, if the more we understand how the activities we do, what we eat, what we drink, lifestyles that we carry, if there is a lot of relationship between understanding, comparing people who have lived longer, those that live longer, and even looking direct interaction, causative action to some of these products, we could help to be able to improve our health by doing, I think this circle here kind of mentions some of the things that we can do, the lifestyles that we can carry to be able to make sure that we reduce our incidence of experiencing cardiovascular diseases. Any question? Any question? I'm going to jump now and talk about, now we've talked about the structure, it will be good now to talk about blood itself. So if you look at blood, how many people have given blood before in the hospital? Anybody has taken? No. How many people has ever given blood as a blood sample? Give me a thumbs up if you have ever been asked to provide blood as a blood sample in the hospital. Nobody? Yeah, like regular labs. Okay, I hope most of you have. And so I just kind of put here like some, let's go here. Do I need to go on full screen? Not necessarily. Let's do this. So this is what I say we would like to know, okay, what is blood made out of? What is its function in the human body? And then we hear the word whole blood, hear the word plasma, we hear the word serum. What are the functions of each of the components that are even blood? We hear sometimes they give blood to people. Sometimes they will give plasma to people. Sometimes they would give what? White blood cells to people. So why do they do that and when do they do it? So we'll kind of look at the composition of blood and the function and the various functions. And then we'll also look at some diseases that are associated with blood. On the right side here, I said the heart pumps about 75 mLs of blood during each contraction. And we said earlier that the heart beats an average of 70 times a minute. And if you do the math, it calculates that the heart will be pumping roughly about 5,250 mLs of blood per minute. I said the entire blood supply is circulated each minute. In terms of function, blood carries, it does transportation. Then you will say, what does it transport? Oxygen, nutrients, waste, carbon dioxide, and chemicals like hormones. It also helps us fight diseases, defense against pathogens. We have white blood cells that help to engulf and destroy microbes, pathogenic microbes. We have others, white blood cells that produce antibodies, which are proteins that help in the process of destroying pathogens. And then we also have cell fragments that help to prevent blood loss during, like you have an injury, so you have blood. It makes our blood to be able to clot, so that blood doesn't ooze out of our body continuously. And then I said it also has a regulatory function, like homeostatic function, in the sense that it transports body temperature. And then I said we have plasma proteins that help to maintain osmotic pressure. You know how we control how stuff gets in, diffuses in, or stuff diffuses out of the blood vessel. And then we have buffers present in the blood. The buffers are substances that would make sure that if your blood is having things that make the pH to be more acidic, then those buffers would make sure that it doesn't get too low, kind of balances. It's like a buffer kind of balances things. It doesn't make it go too low, it doesn't make it go too high. Trying to make the pH of blood around 7.4 most of the time. So in terms of composition of blood, I said you will see now blood has a liquid portion called the plasma, in which is suspended cells and cell fragments. If you take out the cell fragments from the plasma, then you refer to those that portion as serum. Serum is plasma without like the clotting platelets. So which are, you see they call it formed elements. So these are like the cells and the platelets that are produced, as we're going to see, by the red bone marrow. So we have red blood cells, which are also called erythrocytes or RBCs, red blood cells. And then white blood cells, which are also called WBCs, which are also referred to as leukocytes. And then platelets, which are also referred to as thrombocytes. Here, this is like all the sites, referring to cells. Now, I said water makes up about 91% of our body weight. This is messed up because this thing is not moving well. Let me go to presentation mode. Yeah. So if I come here. This is better. So water makes about 91% of our body weight, and then proteins, dissolved proteins like albumins, globulins, fibrinogen, which we'll mention, make about 70%, and then all the other ions, gases, water, products and vitamins and all of the dissolved stuff inside makes about 2%. So where do blood cells come from? All the blood cells that we have, all the formed elements in blood come from stem cells found in the red bone marrow. Red bone marrows are found at the end of our long bones. Like you see here, this is the long bone here. This is at the end of this long bone here. We have we have the red bone marrow here, inside here. And then it has cells that refer to as stem cells. These stem cells are cells that are kind of unique in the sense that they can grow, they could be stimulated to grow to become different cell types. That idea of being able to be stimulated to grow to become different cell types, we call that they have the capability of differentiating. So they are multipotent or pluripotent in the sense that when stimulated by different kinds of factors, some of them will grow to become red blood cells. Some of them will grow to eventually become white blood cells. Some of them will grow to become platelets. So let's say we are running low of red blood cells. A chemical will be produced by our kidneys that will go now and tell these cells, hey, listen, divide. And then when this one is dividing, it would divide and start growing to have characteristics to form a mother red blood cell called an erythroblast, which later on now, this cell that has been committed, it used to not be committed to become anything, but based on that stimulation of that chemical erythropoiesis, which we will talk about, for example, it would now committed now to form red blood cells. And then this erythroblast now will divide, and then to produce cells which when they grow, you see here, this one has a nucleus, this one has a nucleus. But when this one grows now to form red blood cells, when the red blood cells mature, they lose the nucleus, which we see the red blood cell to what it is. On the other hand, if white blood cells, let's say lymphocytes, where these are different kinds of white blood cells, if white blood cells were low or they needed to be replenished or something like that, a different kind of chemical will come here now and stimulate this one to now go and say form this mother white blood cells, whether it is lymphoblast or monoblast or myoblast. Each one of these, depending on need, would go, if this one was needed, it will be stimulated to differentiate to this and then make more of this. If platelets were what was missing, or they needed more of them, there's a different kind of factor or chemical will go and stimulate this one. This one now would grow and differentiate to form megakaryoblast, which eventually will grow to form platelets. So all the formed elements formed because they are created from this to all of this, they come from the red bone marrow. So I say, we've talked about the formed elements. Now let's look at some of the plasma proteins. Plasma, the liquid portion, it has some proteins that are present inside. The most abundant one are albumins. Depending on the amount that is in there, it kind of controls the rate at which things will get diffused in or diffused out. And then the more they can make more liquid to come in. And when more liquid comes in, means more force, the way it would be able to pull more material to come in. And then the globulins, on the other hand, We hear like hemoglobin. This one now is in the red blood cell. We have other globulins that help in transportation. Fibrinogen is one that is also forms threads, fibrin threads, that if you have an injury and your blood is like you are bleeding, this fibrinogen will be converted, this protein will be converted into threads that would block the area where you have bleeding happening. We'll look at that later on. Then when we talk about, let's look at the other one, red blood cells. Let's take red blood cells for example. I said they are formed by, they start from the stem cell. The stem cell will stimulate them if the red blood cells are reduced and your body needs more of them, instructions would come by the production of a chemical from our kidneys to go now and stimulate these stem cells to differentiate to form erythroblasts that will eventually form these red blood cells that look like almost like some kind of lifesavers or something like that, or like donuts with dimples inside. When the red blood cells are mature, they do not have a nucleus. But the cells that form them have a nucleus. In shape, the shape is described as a biconcave shape. And this shape folded inside like this kind of increase their surface area. The round shape makes sure that they are easy to be able to move through the blood vessels. I say they contain about 280 hemoglobin molecules. Hemoglobin molecules made up of a heme group and iron. This is those green sources. So, and then I said the hemoglobin molecule can bind 3 oxygen molecules. So just imagine one of these red blood cells has 280 million hemoglobin molecules that can each bind 3 oxygen molecules. And without the nucleus, it allows this large number of molecules of hemoglobin to be able to fit in there. So you can see now that this structure here, the roundness makes it to be able to move through the blood vessels easy. The lack of a nucleus makes it to be able to hold a large number of hemoglobin molecules, both making the cell very efficient to be a carrier and very efficient to be able to move through the blood vessel. And the surface area increase here because of its shape allows, again, for diffusion of oxygen in and out, because the way the oxygen comes into the cell is by diffusion. And this usually happens in the lungs. This is where the diffusion, this exchange between the oxygen, when the red blood cells move to the lungs, because the oxygen in the air that is in the lungs as we are taking our breath in, the oxygen is higher in the air we are breathing than the one that was being than the amount of oxygen that was present in the red blood cells that were coming to the lungs. So that partial differential in partial pressure would make oxygen to flow in, and carbon dioxide that might have been present will make it come out. So we've talked about red blood cells carrying oxygen. We also know that carbon dioxide is also carried in the blood. So, but how is carbon dioxide transported? When carbon dioxide combines with water in the blood, it forms carbonic acid. Carbonic acid, if you look in chemistry, they will draw this. When you have a chemical reaction, we usually show like this thing that has an arrow on one side. But sometimes there are some reactions that have arrows on one side. That means like half an arrow going this way, half an arrow going this way. They call this reversible reactions. So like in our body, there's some constant change between where carbonic acid is formed, and then the carbonic acid will break down to form. Sometimes will break down to form this, and sometimes will break to form hydrogen and the bicarbonate ion. I said 68% of bicarbonate ion is the way in which carbon dioxide is carried in the plasma. And then 25% of it is carried in red blood cells as carbaminohemoglobin. The 7% is just in the acid form. So you have carbon dioxide in either in this form or in this form or in this form. So if I mentioned the production of red blood cells, imagine a situation where, I don't know, for some reason, whether you're sick or you were bleeding or something like that, or the amount of red blood cells in your system is low. There are sensors that could tell how many red blood cells you have based on the amount of oxygen level. So if the oxygen level in the blood is low, then yes, those systems would inform the kidneys. The kidneys will produce this product, this compound here called erythropoietin. This goes now and goes to the red bone marrow stem cells and stimulate them to produce those erythroblasts that will eventually grow to form, divide, and grow to become more red blood cells. When the increase is normal, but when they are this high amount of red blood cells will enable more oxygen to be carried, and when more oxygen now is now carried and brings the level back to normal in the blood, now those sensors will be turned off and no more instructions will be sent to the kidneys. So it kind of goes into this feedback. It's like negative feedback. You have a situation where more of this, when it's brought to normal, then it tells it to shut down. Erythropoietin is excreted by kidneys cells and moves to the red bone marrow. All cells normally are destroyed. Red blood cells have a lifespan of 120 days, and after that, they go and are destroyed in the liver and in the spleen. How many people have heard about blood doping before? Blood doping. Has anybody heard about that?
I've
Anybody?
never heard of that.
How many people know a guy called Lance Armstrong? Anybody has heard? Yeah. What do you know about him?
He's a cyclist.
It's
He's a cyclist.
Yeah. So they usually have a cyclist competition. There's a famous one in France called Tour de France. This guy has won it like 6 times, where you ride your bike and through different tracks and different hills and this, and then he has won it so many times. So one of the things that they frown up in sports is blood doping. And what is blood doping? I say it is any method of increasing the number of red blood cells to gain advantage during athletic performance. So if you have a way of increasing your number of red blood cells, what does that do for you? It means that more red blood cells means you can pick up more oxygen. You have more oxygen. More oxygen you have, it means that if you have glucose in your blood, it can be broken down through cellular respiration to produce more ATP for you. ATP is the energy that you will need to do your activity. So how does this blood doping thing come? How do they do it? Erythropoietin, we know your kidneys produce it, but we can also make it now in the lab. So some people would buy the erythropoietin and inject it several months before the date of their activity. Then afterwards, when they red blood cells are made, they will go and collect blood from these individuals and store it in bags like this. Now, when they go for prior to the exercise or the competition, they will take everybody and measure their red blood cells number and everything is fine. Everybody kind of averages the same. Now, you go now, the idea is that everybody has around the same average amount of red blood cells. What does that mean? It means that when you go to compete, everybody will be able to breathe in and have a regular amount of oxygen to be able to to uh, to break down the food they have eaten to generate energy. So it's almost like there's nobody at an extra advantage. All you have to do is eat enough, and then um, uh, with the red blood cells, everybody has about the same number. They'll be able to capture oxygen and break them down to produce energy. So the merit of your performance will be a function of how much you can cycle and all of that, not because you have some people are disadvantaged. But what happens when people who do blood doping is when they give their blood, they go and then prior to the test, to the game or to the cycling competition, they measure their blood, everything is fine. But now when they start cycling, sometimes they will stop at certain areas to drink. And during that brief area period where they take a break or something like that, they have each of these cyclists has like a team of their doctors and people who watch physical therapists in their own small booth. When they stop there, they check their bikes and everything. And during that brief period, they would administer back some of the blood that they had taken from that individual. And now that individual now has extra red blood cells compared to the rest of the other person or the rest of other competitors, in which case now they can generate more ATP because of extra availability of oxygen. So that is why people go to do this. But again, it would sound like, well, it makes sense if you just can get more cells so that you can get more energy to be able to do your right. That is a fair thing to do. But this is something that is frowned in all sports. Why? Because if you inject yourself with this erythropoietin, you could produce so many red blood cells that it can make your blood so thick, almost like a strawberry shake or something like that. And when your blood gets so thick, it might be difficult to move along in your blood vessels and it could cause death. So with athletes that have a team of doctors, a team of people that measure and calibrate things for them, might be able to get away with it without dying, without making sure that their blood doesn't get too thick. But if you're in college, high school, or poor or something like that, and you try to buy this stuff and take it in your own privacy, then out of a sudden you took too much, your blood starts getting thick, you could easily die as a result of that. So some disorders related to red blood cells. When you have too few red blood cells, that can be caused from many different things. That general condition is called anemia. When you have a situation where you have some of your red blood cells are not in good shape, structure to be able to do what they are supposed to do, and you have a reduced number of functioning red blood cells, sickle cell anemia, which is caused by a genetic condition, a genetic mutation, where it makes some of your red blood cells, instead of being biconcave shape, they look something like this, tapered at the ends. And then you have just a few, you have just a majority of them being normal, but quite a few having this distorted shape, sickle cell shaped. This genetic condition turns out to be found people in African American descent and other people that are also in tropical areas that have like diseases like malaria. It turns out that even though, okay, first of all, what's the downside of having these cells that are in this sickle shaped? In this round shape, the the biconcave shape allows the red blood cells to be able to move very well in the blood vessel. In this shape, it makes the blood them to clog the blood vessel very easily. And when it clogs the blood vessel, it means that the tissue to which that blood vessel was leading to provide nutrients and oxygen to is deprived of it. Then you start having dead tissue resulting. It also turns out that you need to have, because there are two mutations, you need to have a double of this mutation to be able to have this disease condition manifest. Sometimes, so you could, because we are formed by contributions from egg and the sperm from dad. Sometimes dad might have had the sickle cell mutation in his DNA, and then meanwhile the mom was normal. So that individual was formed just carrying one copy of the mutated DNA gene. In this case, that person is a carrier. But if mom and dad had the same mutations that were defective, then you would actually have this sickle cell disease. It turns out that people who are carriers of this disease tend to do well in areas that have malaria. So the thought about it is that it might be the selection to have individuals that have this mutation, even though in double form, homozygous form is deadly, you could say. But those that are carriers are able to survive in this area, which malaria is a condition that makes people have like chills, some kind of fevers, and can kill them. But people who are carriers of this sickle cell don't have any problem with that. But because of the fact that now populations are moving from different areas with people from these tropical areas with this disease, when they move like to areas that don't have this disease, that mutation doesn't serve them very well, because there's, like in the United States, we don't have malaria conditions. Or if they're very spotty, brought by people who move from these other areas. So it is a disease that affects most of African Americans. And I said about 1 in every 365 Black children is born with sickle cell disease. And then I said 1 in 13 babies are born with the sickle cell trait. Trait means you just have like one of the mutations in one copy of your gene, whether the ones I said that was given to you by mom or by dad. And there's another disease called hemolytic disease of the newborn, which we'll be talking about. I say it's a condition with incompatible blood types that leads to rupturing of blood vessels in a baby before and continuing after birth. We'll talk about that. And then thalassemias, I say it's a group of inherited blood disorders that show themselves as when you have a very few quantities of hemoglobin being produced in your red blood cells. So these are examples of some diseases that are associated with red blood cells. Meanwhile, so we've talked about, first of all, any question about red blood cells before we go to white blood cells? Any question about red blood cells?
I have a question.
Yes.
About anemia.
Yes.
I've heard that so hemoglobin is like iron, or it's made of iron or something.
Mm-hmm.
And that's what causes like when you don't have enough iron in your diet and then you get anemia.
Yes. So if you imagine that hemoglobin is made, it requires iron to be formed, right?
Mm-hmm.
And then if you don't have enough iron, then the molecule cannot form properly.
Mm-hmm.
So if your anemia was caused by a deficiency of hemoglobin of iron, then taking up making your diet being more rich in iron supplements would resolve that problem. But sometimes you could even have a situation where there is this protein here might be the iron might not be the problem, but this protein, which is polypeptide that forms it, is kind of messed up in the way it's not folded properly. So it can be, the whole idea of anemia just means you have a reduced capacity to be able to uh, um, where you have red blood cells that are very few or lack the capacity to carry hemoglobin properly. But iron is usually very, very implicated in most of the anemic conditions that we have. They have a situation where iron is an issue. And the ironically about this is that when they started casting pots, for example, they usually casted pots and made spoons using iron. And it turns out that when people were cooking, some of the iron was being leaching into their food, and it kind of supplied some people with that small quantities that was leaching was supplying people with the iron. But the good thing about this today is if your anemic situation is caused because of not insufficient iron, you can get iron now in iron pills to be able to supplement and give you the right amount that you need.
Okay, thank you.
Any other question? Now let's look at white blood cells. So many a times you say red blood cells are made from stem cells in the red bone marrow because you see red bone marrow. But remember, white blood cells are also derived from the red bone marrow stem cells. And instead of erythropoietin stimulating these stem cells, but there is something called colony stimulating factor that goes to stimulate them to go now to produce white blood cells. I said they can be found in blood as well as tissues. Red blood cells are only found inside our blood vessels. White blood cells are found inside our blood vessels and also outside of our blood vessels. They have a nucleus, the mature ones. See, this is a mature red blood cell without a nucleus, but a white blood cell always they have nucleus. Some of them even have multiple nuclei. Some of them have their nucleus in different shapes. What is their job? To fight infection and are also important to maintaining our immune system to be able to fight other diseases. Some of them would live for days, others will live for months, some of them will live even for years, compared to the red blood cell that has an average lifespan of 120 days. You could categorize them based on whether they have, some of them would have, first of all, whether they have the nature of their nuclei or whether they have granules in them. So if just factoring the fact that they have granules or don't have granules. So you see like this ones here, they have these little granules in them. These ones here do not have granules in them. So granular examples of them would be the eosinophils, the basophils, and the neutrophils. Meanwhile, those that do not have granules, they are granulocytes, they are the lymphocytes and the monocytes. The neutrophils that have granules, they make up about 50 to 70% of all of the white blood cells. They have a nucleus, but it's kind of funky. It's almost like you have a sausage that is connected together. So they're kind of lumpy in lobes. These neutrophils, I said, when you have an infection in your tissue, they are capable of coming out of our blood vessel, and then they will go in there. They have phagocytic properties. That means they will go there and engulf the pathogen and destroy it. Phagocytosis. Eosinophils have a smaller percentage, but they contain also a bilobed nuclei or nucleus. They have large granules again. When you have a parasite infecting you, they can go and destroy it. And then when you have allergy reaction to allergies, they also play a role in responding to allergic stuff. If you have allergies, say to peanuts or stuff like that, the allergy reaction, they play a key role in them. Basophils, not too many of them, have a U-shaped loop nucleus. These basophils produce, when you have allergies, they produce histamine. Histamine will make your blood vessels to dilate. And then all of those things associated with allergy reactions because of these basophils who produce histamines. And then you see why when you have allergy reaction, you take antihistamines to gobble up the histamines that are produced by these basophils. And then when they gobble up these histamines, then they influence that histamine does to make your blood, for example, to make your blood vessels to enlarge. That is, annulled or canceled out when you take antihistamines. Lymphocytes, they make 25 to 35% of all our white blood cells. Conspicuous is that they have a large nucleus that takes up most of the space within the cell, the cytoplasm. We have two different, lymphocytes will develop into two kinds. We have B and T lymphocytes. These are very important in our immune system. B lymphocytes, as we're going to see, is because of where they mature. The B ones will grow and mature in the red bone marrow. The T lymphocytes will mature in the thymus gland. When we talk about our immune system, we'll talk about them some more. Monocytes, they are very few. They are very, very relatively uncommon, but they are the largest type of white blood cells. They have a horseshoe-shaped nucleus. They usually go and stay in tissues. They just go and, like residential guards in the tissue. When you have an infection, they would develop into macrophages. That means they would change into the form that could go and engulf the pathogen and try to destroy it by phagocytosis. So you could see an example. We said you have white blood cells, neutrophils that could squeeze out. This is a capillary. I said they are 1 cell thick. So if you have an infection or with pathogens inside your tissue, they can squeeze themselves out through the 1 cell thick capillary wall and come out there. Or you could have the situation where these ones here that we just talked about here, the monocytes would squeeze out and come out and become macrophages and go and destroy the pathogens. Now, some diseases associated with white blood cells, it said severe combined immunodeficiency syndrome. I said this is inherited. where the stem cells that make the white blood cells do not have the enzymes in the granules that will allow them to be able to fight infection. So you see these, we say these red blood cells that have granules in them, those granules contain enzymes that will go, and when the macrophage has engulfed the pathogen through phagocytosis, these enzymes in those granules would be release the enzymes now go and try to digest and break up the pathogen. But you can have a situation where some of these white blood cells that are supposed to have granules with enzymes lack that capacity. And then sometimes we'll have situations where some of the white blood cells start dividing uncontrollably, which is like, so it's almost like cancer of white blood cells, generally referred to as leukemia. And if you compare the amount from the blood smears, like the ones we saw in the lab, if you compare the amount of white blood cells to red blood cells in a drop of blood, you will see that there's far, far, far more red blood cells than white blood cells. But if you take a normal blood smear and compare to a blood smear from somebody that has leukemia, you will see something like this in the right. This is normal blood, more red blood cells, fewer, and you see these ones don't stain purple because this took the red blood drop, applied this stain to it. But because the red blood cells lacked a nucleus, there is nothing there that would stain, are able to stain the cells. But you see the white blood cells here that have a nucleus, the stain is able to stick to the nuclear membrane and the cell wall, and then you can see these spotted purple structures here. Somebody that has leukemia, when they take the same drop blood from them, look at the amount of, the relative amount of white blood cells they have, a whole lot of them. Then we have another disease called infectious mononucleosis, also known as kissing disease. It's caused by a virus. It would affect the white blood cells, and then it would make people, sometimes they will have sore throats and make them to be tired, affect their lymph nodes and things like that. Then, so now we've talked about red blood cells, white blood cells. Let's look about the other cell fragment called platelets. I said they are cell fragments come from the stem cells. Their stimulation causes the stem cells to form megakaryoblast, which will form megakaryocytes. And then they have about 200 billion are made each day. What is their job is to make sure that our blood we don't bleed, help in blood clotting. I said blood proteins, some of the proteins found in blood, we mentioned fibrinogen, another one is thrombin, are important during the process of blood clotting. So if you compare them to size, red blood cells are smaller, white blood cells slightly bigger, and then platelets they are much smaller than both the red and the white blood cells. Look at the situation, what happens when somebody has, you poke yourself by mistake, and then you puncture, and then what happens? You start bleeding. That is what you see. Oops, blood is coming out. When the blood is coming out, a bunch of things happen. The few platelets that are there, they start coming close to where the opening is at. And then they would produce something called a prothrombin activator, which would convert prothrombin to thrombin, converting this to this. Calcium ions are also involved in this process. And then this thrombin will now go, an existing protein that we find in blood, fibrinogen, in the presence of calcium again, this thrombin will convert it into fibrin. So you have this protein that is normally inside blood. This is also inside blood. And when you have this happen, this activator will come and make this one be converted to this in the presence of calcium. This one goes to convert this to this that is always present in the blood. But look at this fibrin. It now forms like threads like this. So the threads are here now will form like this patchwork here. With the threads there, it's like a net that makes it, the size within the thread are so small that the red blood cells could not drip out. But what you will notice is now when you have a cut, you hold it and press it. When you press it for a while, what is happening is you are allowing many of this cascade here to build up to form these threads. And then when you remove your finger, you go, oops, it stopped bleeding. And then you are looking at it, but liquid might still be coming. Liquid might still be coming through. Plasma might still be coming through. So, but now you see now here that the thread that have been formed would eventually trap all of these red blood cells from coming through, and then eventually you stop bleeding. Some diseases that are involved with platelets could be a disorder in which the number of platelets are too few, or called thrombocytopenia. And then thromboembolism is when a clot forms and then all of a sudden that clot breaks off and start moving in the blood vessel and can go again, just the same way talking about atherosclerosis, a clot can go and clog a blood vessel and cause issues. Sometimes there's a genetic disorder that is caused by people lacking the ability to do this clotting thing. So that a typical genetic condition caused by genetic disorder is called hemophilia. So the problem with hemophilia is that when this person, if somebody that is a hemophilia patient, when they have a small cut, this cascade that we described here, to go all of this to form these threads and stop bleeding, it doesn't work for them. So any small cut will require almost like an emergency room intervention to be able to resolve the problem. So we've talked about blood, for example, its components and where those components are made out of, what blood does as a function to us. And then sometimes now there are situations where people need to be given blood, whether from an accident you lose a lot of blood, now you need to be given blood. And then there is now an encouragement for people to donate blood. How many of you have ever donated blood before? Like in a blood bank, to a blood bank? Anybody? None of you have ever given blood to a blood bank?
I have. I've given blood to a blood bank.
Why did you have to do it? Why did you do it? And what was the experience like?
I did it because there was a blood drive going on by my apartment, and a friend of mine just asked me to go with her. And first they test your blood and they take your blood pressure and things like that, and then they bring you to the back and they take your blood, and then they make you wait for about 20 minutes and also make you drink juice and eat, and then you can leave after that.
Why did they ask you to wait?
Just to make sure that you don't get dizzy from the blood loss.
Exactly. Did you get sick as a result, or?
I didn't know, but my friend did feel a little dizzy, so she had to wait another 10 minutes.
Okay. So donating blood is one of the things that we meaning science has not been able to actually make blood in the lab to give somebody. So the only way in which we can actually, if you lose blood, the only way you can easily have blood is to get blood that has been donated by some other person. And as Tulamri described, the process is safe because the procedure is sterile. Sterile meaning they cannot introduce, it doesn't introduce any pathogens into you. Was it painful? I didn't ask. Was it painful?
Not the process, but the needle is quite big, so it just pinches when they put the needle in. And the person taking my blood had to try a couple times, so I don't think she was as experienced as I was hoping for.
Yeah. So that is, and I'm glad you pointed that out. So the people that go to the phlebotomists, the people who take blood, some of them are very skilled, and their ability is to be able to detect where your vein is, to be able to poke it. And sometimes, if they do a good job, you have a prick that goes in, and then the blood starts flowing. They start collecting it, and then you sit there for a while. Then they will take a certain amount of blood. I said, you will replace the plasma if you are given blood in a few hours, and the cells would be like in a few weeks. Remember, we said the plasma is the liquid portion of the blood, right? Now, they gave Tulam juice to drink. When she drank when she drank the juice, now the liquid portion is easily absorbed and replaced. Then the blood volume almost comes back quickly. And the reason they were asking her to sit down is that when you take such amount of liquid from somebody, the pressure reduces. And if you stand up, you can get dizzy. So when you drink the juice, now out of a sudden, the blood plasma could increase. And then even though it will take a few hours to increase, but at least it will start increasing that you don't get dizzy if you like after the 10 or 15 minutes that you sit down. But I said the one thing that you also benefit from besides doing an altruistic thing to help other people that might need blood is that when you go there and they test for them to take your blood, and before they give it to somebody, they have to test it, to test for blood type, to test for pathogens. The good thing about it is that if you have a pathogen is something that causes disease, right? If you had it in your system and you do not even know about it, they would tell you. And then you could now go back and actually get treatment quickly, because sometimes we have pathogens in our blood at levels that might not be cause symptoms for us for any disease condition. But based on the tests that they use, they can easily see them if they are very, very small copies that are not causing issues. And the sooner you address them to the better as a result of that test. So giving blood is a good thing. It could help save lives for people who need it. And then because also, but don't use blood donation as a way to go have a blood test because it is free. Because as every test, if you have any ongoing disease state, whether you have infections with malaria, hepatitis, or anything like that, you don't want to go and give your blood and let something happen for some weird accident, something happens, and then they give it to somebody who is, let's say somebody was in an accident and needed blood, then out of a sudden they took blood or was infected blood, then it will make their situation double worse. So now that we've talked about blood, the components, some of the diseases that are involved in them, donating blood. And then I also mentioned that when they test the blood, they test it for the blood types. Who knows their blood type? Who knows what kind of blood types they are and what is blood type to begin with? O positive. Pat says she's O positive. Anybody else know theirs?
I'm A positive.
A positive.
They tell you after you give blood.
After the test, right?
Yes.
Great. Anybody else know their blood types? Anybody blood type B? So the idea of blood type is what we're going to go to next, and they mention O positive or A positive and things like that. We'll kind of know what is the A part, what is the positive part, what are they talking about. So I said, some terminologies to understand when we're talking about blood types, which are usually designated A, B, and O blood typing. So on our blood cells, red blood cells, we have on their surface, even though I showed you like this nice round something that was a biconvex concave disc shape, they have small structures on them called antigens. And then, so they would hear a word in understanding this: antigen, antibody, blood transfusion. When you talk about we talked about the blood transfusion here is when you're giving blood to some other person. Antigen, what is an antigen? An antigen is a substance that could cause somebody's immune system to respond to it. Our red blood cells have some structures on them called antigens. And it could, I said, stimulate the immune system. What is the nature of these antigens? Some of them can be polysaccharides or proteins. What is you hear the word antibody. An antibody is a protein that our bodies make to be able to fight an antigen that it has not seen before. An antigen is a foreign substance that is protein or a polysaccharide in nature that would stimulate somebody's immune system to respond. In response to that antigen, which is foreign to somebody's immune system, their immune system will develop proteins known as antibodies to fight off that antigen. So if you take COVID, for example, we said COVID had on its surface spike proteins. Spike proteins, I illustrated in this diagrammatization here. So these spike proteins, if this COVID virus comes into our cell, our body, our immune system will recognize this foreign structure as foreign and respond to it. What will be the response to it? Our white blood cells imagine that this is our white blood cells. When they recognize this protein, they would be stimulated, some of them will be stimulated to produce these blue Y-shaped structures called antibodies. When they shed these antibodies that are specific to this antigen, in this case, the virus, these antibodies will bind on them. When they bind on them, some of our other white blood cells that are capable of recognizing bound structures that have been bound by these antibodies. So when they recognize them, they will go there and destroy them. Antigen is a foreign substance that antigen is a substance that our body finds to be foreign, which are protein or polysaccharide in nature. Antibodies are things that our bodies produce in response to them. So in our ABO blood system, we have two kinds of peculiar antigens on them. And the antigens are illustrated here with this spike with a round blue ring, or a spike with a cone-shaped like a triangular shaped purple color. Let's say we call these ones that are blue A, and the ones that are called are purple B. So for us humans, our red blood cells could either have this, or both this and this. So if you are blood type A, it means that you have only these ones, blue ones. If we agree that we're calling these other blue ones here A and these purple ones B. If you are blood type A, it means that you have only this type on your red blood cell. If you're blood type B, it means that you have only this purple kind. But some people have both, in which case they will have both the purple and the blue ones. So that is with respect to antigens. And some people don't have any of. So it's almost like donuts with sprinkles on them. You say, do you like donuts with sprinkles? Say, no, no sprinkles. Then you are blood type O. I want only these white sprinkles or these blue sprinkles. You are blood type A. I want the blue sprinkles or these purple sprinkles, blood type B. I want both blue and purple sprinkles, you are blood type AB. So that is the way you are the options of the different kinds of red blood cells we might have. These do not have anything to do with the function of red blood cells per se, meaning having an A antigens or B antigens or AB antigens on your red blood cell do not affect the oxygen carrying capacity of the red blood cell. But it helps to identify your red blood cells as yours. So for example, if you have this kind of red blood cells with these A antigens on them, it means that your body has never seen these purple red blood cells with these purple structures on them. So each time our body sees structures that it has never seen before, they get worried. Your immune system, they are like guards that they say, oh, wait a minute, what is this? This doesn't belong here. We have to destroy it. So our white blood cells are capable of detecting things that are things that are foreign. And some of these white blood cells, when they detect these things, they would go and produce antibodies against those things. In this case now, if we have these A antigens on them, if I have the A antigens, so there is no way that my body has seen them. My white blood cells know that I have these. So they will not go to make antibodies to destroy my red blood cells. However, they will have antibodies that could recognize and destroy the B antigens. So if you have a blood type, you would have antibodies opposite to those that you've never seen. So for example, if somebody who has blood type B would have but the A antibodies, blood type A, antibodies against B, they will have it. Somebody that is blood type B would have antibodies against A because they don't have it. They don't have the antigens. If you are blood type AB, you do not have any antibodies against either A or anti-B. Because if you do have them, each time antibodies go and bind to something, it marks that thing for destruction. If antibodies go and bind to anything, there are some cells some destroyer cells that the only way they see is almost like they have night vision glasses, and anything that they could pick up with their vision glasses in the dark, the rest of the cells don't see them. But if they can pick out the things that have been marked, when they see those things that have been tagged by the antibodies, they go and destroy them. So you don't want to have if you are blood type A, you don't want to have anti-A antibodies because they'll go and bind on them. And then all of a sudden, your white blood cells are destroying your red blood cells. That's not a good thing. So people who are blood type AB lack antibodies against A or antibodies against B. Then people that do not have, their red blood cells do not have any of these antigens on them, they have never seen A or B antigens, they would now have both antibodies. They would have anti-A and anti-B. So in this case now, based on what I just told you, if I am blood type A, who can I get blood from? Who can give me blood if I need it, if I'm blood type A? If I am blood type A, can somebody that is blood type B give me blood? Okay, let's say if I am blood type A and somebody that is blood type O wants to give me blood, they would have antibodies against A and antibodies against B in their blood. Do you think these A antibodies will go and bind to my red blood cells and they would destroy mine? So I don't think that would be a good idea. I'm waiting for somebody to make an argument against that. Anybody else? Anybody could argue against what I just said? So when you are giving blood to somebody, right? They are not giving, the blood is minus your white blood cells, okay? So what you do have is if I ask who can give blood to person that have blood type A, if you have blood type O, it means that there is nothing on your red blood cell. So giving this here, your immune system will check it out. Their antibodies will check. Is there any antibodies? Is there any antigen on this red blood cells that I'm receiving that is O? They will say nothing. Then they will welcome the blood. It's fine. So if you are a blood type O, you could give your blood to the person that is blood type A. If you are blood type O, you can give your blood to the person that is blood type B. If you have blood type O, you can give your blood to somebody that is blood type AB. Why? Because there is nothing on top of it that your immune system can see to detect. What about if blood type A wants to give their blood to somebody else? Who can they give their blood to? Who can use blood that is given to somebody by that has a blood type A?
O and A?
So the question is, can A give their blood to this? Okay, let's go from here. Can somebody that is blood type A give their blood to somebody that is blood type B? Can this person give his blood to this person? No, because if they give their red blood cells here, look at this round nature of their antigens that they have. The person has an immune system that has these antibodies on them. These ones that are curved like this, they will recognize this and bind to them, and it will be destroyed. So if blood type A person cannot give their blood to somebody that has blood type B, how about can they give their blood to somebody that is blood type AB? If you have blood type A, you can give your blood to somebody that is blood type AB because they do not have any antibodies against A or even against B, so they could take blood from them. They are missing antibodies against either A or B because their red blood cells have both antigens on them. Somebody that is blood type B can also give their blood here because it doesn't have antibodies against B either. So if you go here, I said the presence or absence of two blood antigens A, B, and the types of antibodies present here, you will have this kind of antigens, you have this kind of antibodies. And then if you have how to remember what each blood type means, I said blood types are named after the protein antigens that are present on them. That's just what I've been indicating. And then there's a simulation here that talks about which ones will bind to what. But I think there's an exercise here that how to determine if blood types are compatible for blood transfusion. If somebody that is blood type A, now if you have antibodies against A, you will have a situation where A would, if you give your blood to somebody that has antibody A, they would not have any problem. I say first consider the antigens found on the blood transfusion recipient who you are giving blood to. Second, consider the antibodies found on the donor who is giving the blood. Okay, so I said if the antibodies in the donor blood, the person giving blood, can be recognized by the antigen on the recipient's blood, then the blood agglutination, blood would clump up, causing a rejection. You do not want to give a red, if you're a donor, you don't want your blood to have on its surface antigens that could be recognized by the person you are giving blood to. Then I gave you some exercises here to do. Can a person with blood type O accept blood type A without agglutination? Why or why not? Why can people with blood type AB accept more blood types than people with blood type O, A, and B? Which blood type is able to be used most often? If we go back here, A blood type O, because it doesn't have anything on the surface, can give blood here to B. Somebody that has blood type B can also give blood to somebody that have blood type A, can also give blood to anybody that is blood type AB. So people with blood type O are usually considered universal donors. It means that they can give blood to anybody. And then if you look at people that are blood type AB, because they lack antibodies against A and against B, it means that if this person blood type A wants to give them blood, it's fine, no problem. They don't have the antibodies like these ones that would have bound these ones. If B wants to give their antibodies here, fine, because they don't have these antibodies that will bind these ones. Blood type O wants to give them blood, fine, because it doesn't have anything on it, because it doesn't have antibodies against either of these. So blood type AB are considered universal acceptors of blood. They will get blood from anybody, and blood type O, universal donors. They can give blood to anybody. Try to remember that. Then the other question that you were mentioning is we talked about A positive, B positive. What does all that mean? So besides the A and the B antigens on the blood, there is another factor or another receptor they call that people have. You can have it or you don't have it. If you have it, they call it is positive. If you don't have it, they say you are negative. It's called the rhesus factor. People who have it, I said, besides you could be blood type A, and then in addition you have this other receptor. It's called you are positive. So you are A positive if you have the rhesus factor. You are A negative if you have the A receptor, the A antigen, but lack this rhesus factor. The rhesus factor itself could elicit the rhesus antigen can also elicit the response via immune system. So if somebody had blood type A and then receives blood from somebody that is blood type A as well, but they had the rhesus antigen on them, they would also react, their immune system will react not because of the A antigen they have, but because of the rhesus factor that they have. This situation is a little bit even more critical, especially with newborns, when in situations of pregnancy. We'll look at it. I said, sometimes if you have a couple who are about to have a baby, and let's say, for example, the moms, because normally the baby is coming from the egg that was given by the mom and the sperm that was given by the dad. Let's imagine a situation where the mom's blood cells have the positive, did not have the rhesus positive antigen on it, but the dad had it. So they have the egg and the sperm. The sperm has the rhesus. Now out of a sudden now the fetus that is developing, because of the presence of the contribution, the rhesus positive contribution from the dad, the fetus is rhesus positive. Fetus, which is eventually going to develop to a baby. But remember that mom is rhesus negative. So the fetus grows, and then it's time for delivery. They go now, and the baby is coming out. Some of the baby's blood comes and then comes into contact with the mom during delivery. And then out of a sudden, the mother's immune system sees this rhesus positive. And then her immune system develops antibodies against this rhesus positive because of the exposure. Everything will be fine, the baby will be born, but out of a sudden, the mom's state changes from being rhesus negative, meaning that do not have any antibodies against the rhesus antigen, but because of the exposure of her first baby during pregnancy to the rhesus positive blood of the fetus, she developed antibodies against the rhesus positive antigen. Now, what would happen if they want to have the second baby? If she's having the second baby, hopefully with the same husband, then when the husband normally contributes his sperm that has the rhesus positive, and the mom contributes her egg that is rhesus negative, as the fetus is growing, because when the fetus is developing, there is some connection between the fetus's blood and the mother's blood. During this connection in the placenta and stuff like that, guess what can happen? Some of these antibodies that are against these rhesus antibodies that the mother has now could pass through and go and start fighting the fetus while it is developing. So I said, the antibodies can leak across the placenta and attack the developing fetus during the second pregnancy. And it could attack it in such a way that it can kill the fetus, or make that in such a way that when the fetus grows and is born, it could grow and suffer from mental retardation, or can even cause death after it is born. So in situations like this, the only way to have this couple, mom being rhesus negative, dad being rhesus positive, to continue having more babies and avoiding this situation, which is referred to as hemolytic disease of the newborn, is that each time when the mother has a baby and she is exposed to this rhesus positive from the fetus, she receives a shot that would be able, some treatment that will be able to destroy any antibodies that are trying to form in her. I said here, rhesus positive red blood cells leak across the placenta into the mother's bloodstream. And then B here, mother forms anti-rhesus antibodies that cross the placenta and attack the rhesus positive developing fetus. To be able to resolve this, rhesus negative women are usually given an injection of anti-rhesus antibodies no later than 72 hours after birth of a rhesus positive baby. These antibodies attack fetal red blood cells in the mother before the mother's immune system can make any antibodies. This will have to be repeated each time if the rhesus negative mother has another rhesus positive baby in case of later pregnancies. So now we have spoken about the A and the B and the AB and the positive and the negative. Positive and negative is with comment as to whether you have this anti-rhesus, have this rhesus antigen or not. Any question about this? Rhesus positive and rhesus negative. Any question about the rhesus positive or all the antigens that we have on our red blood cells? So the ending I was ending on this slide was basically to tell you that, you know, this is how, if you look at it now, we're going to, I said, we talked about the cardiovascular system. This system works along with all you could see I was mentioning how it works with our respiratory system to be able to supply our cells by the gaseous exchange that happens in the lungs. It works with our digestive system because it will carry nutrients from digested food to the cells. It works with our urinary system because when our kidneys would remove things from blood, waste from blood, like to form urine, which is now we flush out. It works with our muscular system because our muscular system is there to contract and press on the blood vessels to be able to move blood around. With our nervous system, again, because when our nervous system is able, for example, to pick up monitor low concentrations of oxygen, for example, and give instructions for more blood for more red blood cells to be made, or to increase our blood pressure to pump blood more. Endocrine system, so our blood vessels transports. Endocrine system is the system that looks at the hormones that are being transported and produced by our body. Hormones, as you're going to see, are chemical signals that go around in our body and makes our bodies to do certain things. That these hormones are carried in blood. And then our lymphatic system, again, as we are going to see, some of the white blood cells that are present in this system go into our cardiovascular system, carried to distant areas to help to make sure that we don't have any issues. And excess blood liquid within our blood, some of it will leak into the lymphatic system to make sure that the blood pressure kind of stays where it's supposed to be. Our skeletal system is also involved in our cardiovascular system. Why? Because we see that our red bone marrows are the ones that produce our red blood cells and things like that. So if you look, we have different systems each system made of a set of organs working together, but all of these systems work together to keep us healthy and alive. We are going to touch on all of these systems, not in depth, but touch on all of them as the class migrates, as we progress in the class. So does anyone have any question about the cardiovascular system? Any question? The time now is about 11:54. Let's take a 10 minutes break, bio break. If you think of any questions, you can come up and we talk about them. But and then afterwards, actually, before we go for lunch, let me just take 5 minutes here and talk about the study guide, so that since we'll talk about what is it that we are going to be doing for where are we at here? Let me see here. Talk about the study guide, since the study guide has it contains everything that we have dealt. Let's talk about it just briefly and then go for lunch, come back and then talk about something else. Let me just see this here. So I'm going to share. You should have access to this now. Do you see my screen?
Yes.
Yes.
Okay, I'm going to make this slightly bigger. Anyway, you should see the same thing I'm seeing. So we started. So it's almost like the first 6 chapters is what we're going to be talking about. And the way I made the study guide is in such a way that I ask you some basic questions as a guide because based on the questions I'm going to give you. So if you can answer these questions, then when I present the questions in a multiple choice format, then you'll be able to pick out the right answer. What is, for chapter 1, what is biology? What are 8 characteristics of living things? How are human beings related to other things? How do we classify life? So I do the same thing here. So if you have the patience to be able to take time and answer these questions, or if you are studying with somebody else, you could quiz them to ask them these questions, let them be able to answer them for you, to you, and then so that you can see whether you know the material. And then the chemistry of life, just as on the basic chemistry, what is an atom, how are atoms organized, the structure of an atom and the subatomic particles, or how are DNA and RNA similar, talk of all those macromolecules. And then we went and talked about cells. What is a cell? What is the cell theory? Why are cells small? What are the differences between prokaryotic and eukaryotic cells? So I asked you these questions. It should serve as a guide on you answering them as you read along. Look for the answers. And then I'll be presenting these answers, these questions to you in a multiple choice format. Then this is the cardiovascular system that we just talked about. Some of these questions are the same questions that I said that by the time we are done with this, you should be able to know this. What is the cardiovascular system made up of? The heart, blood vessels, and all of those things. So what are the functions of the cardiovascular system? That is basically what is it that we discussed as we're talking about this. And then we went to talk about blood as we just completed. What type of tissue is blood? Connective tissue. What are the blood components? What is plasma? What is it made out of? Name 3 formed elements. So all of this. So I hope that these ones here would make it easy for you to focus as you study and then know how to answer them. Because if you I could have asked you these questions like this, and then you write the answers, but this time it's going to be multiple choice. I've given you the questions. I will just give them to you in a multiple choice format. The beauty is I will give you the correct answers as options. Not so beautiful is that I'm going to give you some distractors in terms of the answers for you to be able to pick out the correct one from the mix. We have labs. We have labs that I've given you. We have done a lab. We did a lab in the first lab that we did was online. The second lab that we did was in person. The third lab that we have done was we did it online. And today's lab, we're going to do some of it. We're going to do some of it in the lab today. And I've also assigned you also did some lab, the lab that today will be talking about who can give blood to who and things like that. And then we'll also to talk about how enzymes work and why is it that enzymes are proteins, what are the things that would influence enzyme activity and all of that. And then you will be all set for the exam. And so when we come back next week, I'll basically ask you if you have any questions. So between now and next week, if you have any questions regarding this, you can ask me. When we get to the lab, you have any questions, you can ask me. I'll be able to address it. And then when we come next week, no questions, I will assign the lab. Then you have for a couple of days, then you're able to do it. Any question about the study guide?
Not about the study guide, but about the exam itself.
Okay.
Is it online or in person?
It's going to be online.
And then do we take it in the lab or at our own time?
At our own time, at your own time.
Okay.
But it will be, I'll give it a window, like 2 days to take it, within 2 days to take it.
Okay. And then also the lab quizzes are going to be online or in person?
But this one is going to be online as well.
Okay. Thank you.
Okay. So it's actually, I'm going to stop sharing now. It's about, it's noon. Let's go take lunch and come back at a lunch break and come back around 15 minutes, 12:15. And then we will talk about the digestive system and start talking about the lymphatic system. Okay, I'll see you shortly. Welcome back. Can you hear me?
Yes.
Yes.
Okay, let's talk about the digestive system. And then by the time that we are done, I think we should be able to talk about, where is my cursor? Why is this not moving? Okay. We'll look at as in a system, we're going to look at the structures and the components that are involved in the system and know what each one does. We're also going to talk about the various food types, categories that we normally eat in terms of carbohydrates, proteins, and fats, and then look at how they are digested and absorbed, and then look at the nature of all of these components, whether they are proteins, lipids, and carbohydrates, what they are needed for, and then talk about other nutrient types, essential and vitamins, And then we'll talk about some disease conditions that are associated with nutrition and digestion, talk about cholesterol, and then talk about diet to some extent, and then talk about some disorders that are associated with diet. But I think just for a quick review, let's take a, let me see here, and put this on where is my cursor? I'm not seeing it. Okay. Let's see if we could watch a quick overview here of the system. I'm going to start playing. Let me know if you can see it. If you can see it, somebody should just give me a thumbs up if you can hear and see what it is about.
Primary functions of the digestive system are the breakdown of food, called digestion, and absorption of nutrients. Digestion begins in the mouth, where the teeth break food into smaller particles during mastication. Salivary glands located near the oral cavity secrete saliva, which begins chemical digestion and keeps the food moist. As food is swallowed, the soft palate blocks the upper pharynx to prevent food from entering the nasal cavity, and multiple voluntary muscles in the face, neck, and tongue contract, pushing food particles through the pharynx. The food passes over the epiglottis, which prevents food entry into the respiratory system, and then into the esophagus, which connects the pharynx to the stomach. The one-way movement of the food mass, now called a bolus, is controlled by wave-like involuntary muscle contractions. This movement is known as peristalsis. The bolus now enters the stomach. Folds in the stomach wall, called rugae, allow for expansion as the stomach fills. Stomach cells secrete hydrochloric acid, pepsinogen, and various regulatory hormones that chemically digest the bolus. Muscular contractions in the stomach churn its contents to further break down the bolus and mix it with stomach secretions to form a thick liquid called chyme. Chyme exits the stomach through the pyloric sphincter and enters the small intestine, the major site of nutrient absorption. The small intestine consists of three parts: the duodenum, jejunum, and ileum. Bile from the liver and digestive enzymes from the pancreas empty into the duodenum to aid in digestion. Absorbed nutrients pass from the lumen of the small intestine into blood and lymph. Chyme not absorbed in the small intestine enters the large intestine. As it passes through the cecum and ascending, transverse, descending, and sigmoid colon, water and salts are absorbed and chyme is converted into feces. The rectum stores feces until nervous stimulation initiates the defecation reflex, resulting in elimination through the anal canal.
Excellent. So that is a quick overview of our digestive system and process. And let's take a quick tour of what is involved in the process. So the goal of digestion is we have this food material that we want to break down into small portions that can be absorbed by our body for use or to further break down to release energy for us. So we have the main path that was shown there. It comes from our mouth. It goes through the pharynx, goes through the oesophagus. From the oesophagus, it goes into our stomach. From the stomach, it goes to this curved section called the duodenum here. And then from the duodenum, it goes to the small intestines. From the small intestines, it goes to the large intestine that moves up, transverse descents, and then during all of these different things are happening, undigested material is defecated out. Some ingestion, taking food into our mouth, digestion, breaking down the food, and it could involve breaking it down into smaller portions, mechanically or chemically. And then mechanical will be when we break it down with our teeth, when the stomach is churning it. Chemical would involve the breakdown using enzymes. The goal is to end up with subunits that can be absorbed by the body. The food is in continuous motion after you have swallowed it. Then it keeps moving until the point at which along the way the digestion is happening, and then along the way the absorption of material, digested material is happening, and then towards the end the undigested material stays in a rectum area until when it is defecated. We talked about absorption, movement of small subunits into the gastrointestinal tract, movement across them, and then through the walls into the blood vessels, elimination, the removals of undigested molecules. Same view of the same thing, but here also included is the functions of what is happening during each of these steps. In the mouth, we have teeth with dentition that is designed and suitable for the kind of food that we eat. The teeth is to cut the food and grind it up, mastication, and then the tongue is there. And then within the mouth too, we have salivary glands that are opening into the mouth to mix whatever food is, the content with saliva, which has enzymes to start digesting some portions of food like carbohydrates. So when we chew it and then it forms this bolus, we swallow it. Swallowing it is a kind of an interesting show because as we are swallowing it, we have a structure here called the epiglottis. The beauty of this structure is when we swallow the food, it pushes the epiglottis to close the opening that is going into our trachea and allow the food to move through the esophagus. They describe the mechanism as which the food moves through this esophagus, called this contraction that keeps squeezing the food as it moves along, referred to as peristalsis. As we lose control to the point at which after we swallow the food, the movement of the food is just now involuntary. We cannot control when you have swallowed it, start moving down beyond that point. We cannot tell the food to keep moving or move faster or slow down or bring it back up. So it falls into our stomach. In our stomach is kind of folded, as they showed you, is folded in such a way that it can expand when more food comes inside, but also interesting in the walls of the stomach. We have this gastric pits that will secrete gastric juice. Some of it will secrete hydrochloric acid. Some of this hydrochloric acid is crucial for the functioning. Some of the enzymes that will start the digestions of proteins. Inside the stomach churns the food as mixing up with these gastric juices. The end result is it becomes now this slimy material now called chyme or chyme, and then it will now move through to the first portion of the small intestine. This portion here that is kind of like an inverted C called the duodenum. And then from there now, this portion, the duodenum, has a connection from the pancreas that leads to it, because pancreatic juice, as you saw, comes and mixes up with the food that has left the stomach, and then there it continues further digestion. Then it goes to the small intestine, where completed digestion is complete, but now absorption starts as well. Like I said, in the small intestine, the chime with digestive enzymes for final breakdown and absorption of nutrients and molecules into the body, into the blood vessels, and then it secretes digestive hormones into the blood. Now, in the large intestine, as the food exits from the small intestine, mostly the absorption of water and salt happens in the large intestine, and then until the undigested material is now defecated. Remember, we don't have complete digestion of our food. As the food is transiting, not everything that we eat is totally broken down. There are certain things that we don't even have capacity to digest. Like, for example, cellulose. We don't have, you'll see that we are limited. When you eat like lettuce, eat spinach, yeah, we will digest some of the stuff in there, but the cellulose part of the food that we eat in our vegetables, we cannot digest it because we don't have enzymes for it. I mentioned a couple of accessory organs here, the liver and the pancreas. The liver plays a major role because it, first of all, it stores nutrients like glycogen. It also produces bile, which is required for the emulsification of fats, basically making we've eaten and breaking them down into smaller globules to allow to increase surface area for reaction of lipase enzymes that will act on the fat. And then it also, we have the pancreas that, I said, produces pancreatic juice. We know the pancreas produces insulin and glucagon, but it also secretes pancreatic juice that contain enzymes that will complete digestion. So now let's look at some of the cross section and the transverse section of the digestive tract. If you look at it here, we have an innermost layer called the mucosa. I said it has mucus that protects the lining and also produces enzymes. Then you have a portion of the submucosa, the layer that is beneath, that is a loose layer of connective tissue with blood vessels right here. And then we have the lymphatic vessels also present there. And then you have vascular, not vascular, we have smooth muscle cells here that enable the contraction. And as we saw smooth muscles, the contraction is controlled, is autonomous. Autonomous contraction means that we are not aware of the contraction process, but information comes from our nervous system directly influencing how they will contract. The outer lining, they refer to it as the serosa, the outer lining. And then you see all bits folded inside here. You see all these foldings that are also inside here, all to increase surface area. So if you look at what it does, what is its job? Have things that would secrete enzymes in the gastric, has blood vessels that would absorb. It is folded to be able to expand. If you look at our mouth, I said our teeth, kind of most living organisms, animals, they have dentition that is suitable to inform what kind of diet they normally eat. If you look at us, we have different kinds of teeth in our mouth. Here we have incisors. Incisors, they have at the tip of them, it's a little bit sharp, flat. So it's like they use for cutting. Then we have a pair of canine teeth below, and then a pair above in our upper set of teeth. Then following those, we have premolars and molars. So if you look at it, we have how many molars? Molars have flats, and premolars, they have flat surfaces that are used for grinding. For those for cutting, we have 4 of them, a total of 8. And then we have only like what? four canine teeth. Canine teeth are usually used for tearing. Dogs have more of them. Dogs don't have, molars and premolars because they tear their stuff and they eat it, and they eat it. So if you look at it, if you're going to cut stuff, we cut things like vegetables. We also grind stuff. But if you look at cows that are able to also grind stuff have molars, and they have incisors for cutting. So they eat a lot of grass and stuff like that, suitable for that. Dogs, cats, they have canine teeth, suitable for eating meat stuff. But we have some teeth for cutting and for grinding. We have just a couple of 4 teeth for tearing. So it probably is almost suggesting that we should be having less meaty tearing stuff, but more cutting and grinding kind of foods, which should be more vegetable. But generally, us humans, we are omnivores, meaning that we eat both plants and animal sources for food. Talking about the teeth, we have 32 of them in adults. And what we normally see outside in terms of structure of the teeth, what we normally see is the enamel that is clearly visible, the white part that everybody likes to keep white. But when we are growing up, before we start ending up with 32 teeth as an adult, we have 20. When babies are growing, they have 20 deciduous teeth. It means that they would eventually fall out and then be replaced by more permanent teeth. The greater part of a tooth is embedded, is more within our jaw. So you have a portion here that is visible called the crown, and then the part that is the root now is more embedded in the jawbone. Then we have this gum here that separates the top part, the enamel, and this is the area where we have issues with cleaning up to make sure that things don't get trapped in it. And there are situations sometimes which you would hear, you have a food a bacterial infection. Bacteria can come in your gum here and infect. If you don't brush your teeth, what will happen is that if you have food particles here at night, you close your mouth, anaerobic respiration happens, and then the end result is some acid is because carbon dioxide is produced. You have acid that is produced, then it can damage your enamel, and then you have issues. So sometimes you have situations where you have an infection and then right up into these cavities here, and now they would have to do a root canal to drill and access it, use either UV light to destroy it, or they would find some way to give you antibiotics to deal with it. But the pharynx here, if you look at the structure again from the mouth, we have salivary glands that squeeze saliva into it. We have after you have chewed your food, mastication, then you have your tongue that would press the chewed food against your upper palate, which now forms the food bolus. As we saw, it moves down, pushes down on the epiglottis. I said it closes the trachea, allowing the food to move through the esophagus, opening up only. This is the bolus migrating. And then now, as it gets into the stomach, there is a sphincter here that would prevent food from coming back into the esophagus. The stomach usually has a acidic pH around 2, and you don't want material that is within from the stomach to come back into your esophagus, or else you would have burning sensations, because the walls of the esophagus are not equipped to deal with the acid nature of the food that is already in the stomach. So when does heartburn occur? I say it occurs when acids from the stomach, for some weird reason, will try to move back into the esophagus. They cause that gastroesophageal reflux, and sometimes it will cause that burning sensation. ways of people who have this, you don't eat stuff that has a lot of fatty meals, too much of it, don't overeat. And then it's good to eat smaller portions because you don't want your stomach to fill up too much because of it going backwards. And then when you eat it, you don't exercise too much. That can also cause that to happen. Within the stomach itself, I said the stomach made up of multiple layers, a layer that has muscles, smooth muscles to allow it to be able to contract, to enable the churning of the food while it is in there. We have gastric pizza that secrete gastric juice. The gastric juice contains pepsinogen, which is going to be an enzyme. In the presence of hydrochloric acid, it gets converted to pepsin, and this starts breaking the proteins into peptides, into shorter protein fragments, start bringing the peptide bonds within the proteins to start cutting them. The walls of the stomach have mucus that prevents the effects of low pH from the acid affecting the cells that are, within the walls of the stomach. We talked the last time when we were talking about the scientific method, we said sometimes we have a bacteria called Helicobacter pylori that could sometimes erode the walls of the stomach and then cause wounds in the stomach. And in this case, it will be treated with antibiotics. The stomach, as it empties, after it has churned the food that is inside and started the digestion of proteins, it forms this smeary or this material that empties into the called chyme that is now goes into the next step called the duodenum. Depending on the food type, it might stay a certain amount of time, but the durations are usually about 2 to 6 hours within our stomach before it moves on. But remember, chemical digestion actually starts from the mouth because digestion of carbohydrates starts from the mouth. The salivary juice that are produced contains salivary amylase that starts acting on carbohydrates. And in the pH in our mouth is basic, that means above 7, allowing for this enzyme to function. When it moves into the stomach, whatever we're chewing has moved into our stomach, because this salivary amylase likes a high pH to operate within, its function stops. If your food that you had eaten had contained proteins, then the pepsin that is found from the gastric juice starts acting on the proteins. Now, whatever is moving from the stomach to go into the duodenum, that second compartment, requires the enzymes to mix with enzymes that are coming from the pancreas. In this case, those enzymes work at a higher, they work at a higher pH. So material that comes from the pancreas has buffering material, solutions that will make sure that it changes the pH from the acidic pH of around 2 to an increased pH that will allow for the rest of the other enzymes that are contained in the pancreatic secretion to complete the digestion. I said, if you look at the small intestine, it's about 8 feet long. The first part of it, which we call the duodenum, is followed by another one called the jejunum, the ileum, where most of the absorption happens. We also know that bile is secreted from the gallbladder into the duodenum to make sure that if whatever we're eating had like fats, then it would break down this fat into smaller fat bubbles, or called globules. And the goal of doing this is to make sure that it increases the surface area on which the enzymes lipase can go and act on the fat to break it down. Where is bile produced? Bile is produced by the liver and stored in the gallbladder, and then connects to the duodenum. As a digested food is absorbed through a large surface area made large because of the folded nature of the inside part of the small intestine that forms these finger-like projections called villi. Villus is one, many of them are villi. In terms of absorption, digested material, proteins are ultimately digested to amino acids, carbohydrates to sugars, fats to fatty acids and glycerol. Both amino acids and sugars would get into the blood vessels. This is an example here on the right here. You see here, this is one villus, a cross-section. Within the villus, you can see here a blood vessel, the arteriole that is bringing the blood, and the one that is leaving the vein, this venule that is leaving. But inside there is a lacteal vessel. The amino acids and the sugars would be moved into the blood vessel. The fatty acids and glycerol will move into the lacteal vessel, which is part of the lymphatic system. A bigger appreciation of so again, the arteriole, or the small blood vessel that is bringing blood to the villus, and the one that is leaving. And then again, the lacteal vessel, part of the lymphatic vessel. Now, in terms of this, again, it's like a summary slide. We start with carbohydrates, basically a chain of monosaccharides. Generally, all the enzymes that act on carbohydrates are referred to as amylases. So you have salivary amylases, means enzymes that are in saliva that break carbohydrates. You have pancreatic amylases, enzymes that are secreted by the pancreas that would act on the carbohydrates. These combined efforts of breaking of the carbohydrate chain into the monomers is done by this set of enzymes at different locations. The ultimate is to create monosaccharides that will be absorbed into the blood vessel. Proteins held together, basically a chain of amino acids held together. So you have peptidases, the way you refer to generally all the enzymes that would act on proteins. Peptidases, they break peptide bonds. So we started by pepsin in the stomach, and then we'll continue to trypsin in the duodenum. The goal again is to break them into amino acids that will ultimately diffuse into the blood capillary. Bile salts, from bile, they break down fats into globules. Lipase acts on them, breaking the fat into fatty acids and glycerol, and they eventually absorbed into the lymphatic system. This is a good summary slide that indicates the various enzymes, what produces them, where they act, the desired environment in terms of pH under which they are operating in, and then the products that results from the digestion from the substrate. It shows here pepsin digestion, nucleic acid fat digestion, and then it's a good summary slide. It's a good one to remember. I've mentioned to you the function of the pancreas, the function of the liver, and that of the gallbladder. Besides the digestive enzymes produced by the pancreas, we know that it also secretes the bicarbonate material that goes now to counteract the low pH from material that is coming from the stomach. And in this higher pH allows for trypsin, lipase, and pancreatic amylase to be able to operate it. The pancreas also produces insulin and glucagon. Insulin will convert excess amount of glucose into glycogen for it to be stored in our muscle and also in the liver. When there is less glucose in our blood, glucagon also produced by the pancreas goes and converts stored glycogen into glucose to bring up the levels as desired. I said the liver has made of many lobules, and then it lies right underneath the diaphragm, filters blood from the gastrointestinal tract, removing poisonous and detoxification, or removes iron, vitamin A, D, E, and K, these are fat-soluble vitamins, and then also vitamin B12 from the blood and stores them. I said it's a storage site for glycogen. And then I said it makes plasma proteins that will help to regulate cholesterol as it makes bile salts. So when it makes the bile is then stored in the gallbladder, which will subsequently be delivered into duodenum as needed. It breaks down hemoglobin in forming the bile. Some liver disorders that we might know, sometimes the liver might become inflamed as a result of an infection like hepatitis. They have different strands, hepatitis A versus B and C, but it could also result, if you have this infection, it could cause to liver damage, it can even make the liver cells become cancerous and could kill somebody. Sometimes the liver can become fatty, have more fat, and when it's more fatty, it's not able to do its job, like they call it cirrhosis of the liver. And then when it becomes fatty, then it would have eventually can form scar fibrous tissues. When people drink a lot, they usually have this condition that can result with the liver becoming fatty and have cirrhosis. And it could cause liver failure. And I said the liver cannot be regenerated, and it could cause problems. All of this action on food is present in the duodenum, pancreas. Now it's time for you to secrete pancreatic juice, or food is present in the stomach. Now pancreatic pits, gastric pits secrete pancreatic juice. We have a set of signaling communication that communicates from the brain, or that senses the presence of this food and tells these various organs to secrete material. And then when their effect has been done, it also signals and tells them to stop. So you would have things like secretin, cholecystokinin, gastrin. All of those are signaling molecules that would drive some of the activity of these various organs. The large intestine, you could divide it into the ascending part, the transverse part, the descending side, and then you have the rectum here. What is happening here mostly is material that was not enzymatically digested has to at some point be delivered here. When it's delivered here now, excess, if the body needs more water and salts, they will be absorbed along as the food tracks along here. In the rectum, they just stays, the material stays in there until it fills up and then signals for you to go use the bathroom to defecate. Some disorders of the colon and the rectum would have diarrhea, where you have loose stool, I said increased peristalsis, and water is not absorbed properly. And then you would have a situation where your poop is liquidy, and sometimes infections can cause this. Constipation is almost like the reverse, where you have feces is dry and hard, it's difficult to defecate, and maybe because of less amount of fiber in your diet and or lack of drinking enough water can also cause this. Then you have inflammation of the blood vessels around the anus, could cause a condition called hemorrhoids, could happen due to chronic constipation. Sometimes this is a condition associated with pregnancy and aging, and worst of all, when people engage in dangerous acts like anal intercourse. Sometimes the mucosa that forms sagging and forms pouches in them. And then when the foot goes in there, it doesn't move along continuously. So it like goes and gets stuck in there. And then when it gets stuck in there, some of the bacteria in that foot that is stuck there would divide and increase in numbers and could cause issues. We call it diverticulosis. And then there's a condition, irritable bowel syndrome, just the syndrome. That means you have a situation where you have uncontrolled contractions of the muscles of your GI without really in a coordinated fashion. So the normal coordination that is supposed to happen, food is there, it's time to contract, it's time to stop, it doesn't happen. The result is that you have chronic diarrhea and abdominal pain. And it is something which they don't really understand yet properly what can be causing irritable bowel syndrome disease. One, you could say there's some conditions they will call like Crohn's disease, ulcerative colitis is some of these irritable bowel diseases. And there have been no, they don't really have a way of curing this yet, but one of the ways that they have seen some results, sounds weird, is doing fecal transplants. And it is with the thinking that the microbiome, because we have a lot of different kinds of bacteria in our GI tract, especially in our stomach, we don't really know. We know the different types of bacteria that are present, but we don't know the amounts of each type that allows for to create a normal condition. So not knowing whether bacteria A, you are supposed to have this level, or bacteria B or C, you are supposed to have this level, is very difficult, even though you might know the different kind of bacteria, they call them flora, that is in your stomach, that your stomach is made out of. It doesn't help solve the problem because the people that have been mapped they will have the right representation of bacteria, but they still have the syndrome, irritable bowel syndrome or disease. So, but what they have seen is that if they take fecal material from somebody or people that have a good representation and they transplant it, I will show you in the next few slides, they dehydrate it and put it in a capsule form, and somebody swallows it and it goes there, and if reestablishes the bacteria representation in there, sometimes they've been able to see relief as a result of that. Sometimes you would see growths in the walls of like the large intestine, like polyps, or some of those polyps, like finger-like small projections in the inside walls of the large intestine. When they see them, some of them might be benign, means they don't do anything. Some of them could actually grow to become cancerous, which can come from colon cancer, which can be a big deal. Now, we've talked about digestion, what is being digested, what are the various areas where digestion happen, enzymes that are involved, the products that are produced from the digested food types, and where absorption happens. And now we've also mentioned some of the disorders related to some of, to the, like to the colon that we just talked about. The part that I would like us to talk about next is about weight. We have a standard way of looking at weight. We have known that when people who are overweight, when you say overweight, what does that mean? In recent times, we've been saying that they look at their body mass index. It's used to estimate how much fat somebody has as opposed to muscle. And I said, for women, the body mass index for women and men is slightly different. For men, it's between 8.5 to 24.9 for healthy men. And then I said, considered greater than 30 is considered grossly overweight. Now, this is there's been some revision in this thinking that sometimes they don't really take account into some of the genes that are involved in the individual. And there's been some revisions that are being made. If you read current papers, you will see that some people who are thin or they have a low body mass index, and you see that, they are not obese, yet they still have some pathologies related to that. But some people who with a large body mass index, and which will be termed overweight, they are still healthy. But we have also seen that having weight associated with fat has also been associated with a lot of many diseases. So the idea is to make sure that you have your weight is not too much as a result of how much fat and you are carrying on, and where the fat is deposited on, distributed in your body, also has been seen to be correlated to different kinds of disease condition. I said the part that this body mass index doesn't take into it is how much exercise the individual is doing and how much the bone structure is, because some of these things, exercise, lifestyle, can influence, even though you might look big, but because of the amount of exercise and because of your bone structure that can account for it, then it might skew to the fact that even though you look big, you are not having any disease conditions. There is a chart here that tells you about, kind of gives you an idea of, I think your book has one too, talks about how tall you should be, how much you should weigh, and then which one is healthy weight, which one is overweight, which one is obese, and things like that. But I would defer to your doctor. If you have any concerns about this, you could have discussions with your doctor to be able to set you straight, to give you the right perspective on where to go. I said the reason that this weight is of, just as I was talking to you about cardiovascular diseases, why we should care whether people are having good cardiovascular health, it begs the question, why should we also care whether people are obese for the similar reasons? One observation is that for the past 20 years, people that are considered overweight, the amounts have doubled in the US. And well, more in the eastern, southern part of the country than to the western part. I said the US has an average of 1/3 of its adult population being obese, and it is now very prevalent in children and adolescents. What can be causing this? Is it lack of exercise? Is it the kind of food we eat? Because in developing countries, the incidence of being overweight is not as, they don't have that issue compared to what we have here in our country. And in developing countries too, we don't have situations as prevalent like for irritable bowel syndrome kinds of diseases like we have here. Some people are attributing this disparity that maybe it might be related to the fact that in developing countries, they don't depend a lot on processed food as we do, and they eat more in what is in season compared to us here. Here, I can decide and say, I'm going to be only on the diet of steak, French fries, and I can eat mangoes. So, and my source of this food is going to be from Safeway or Albertson, and I can get that all year round. In developing countries, they have seasons. They have like a wet or the dry season, different crops or different food types become available during each of those season types. And then most of the people who live subsistently, meaning they cultivate or they grow their own food and eat, or go to the local supermarkets who sell what is in season, they depend on those kinds of various kinds of food, diversify the kind of food they eat. And each of those foods will also come up with their own bacterial flora that go and reestablish in their stomach, so that the idea is most likely that this eating in season allows them to also replenish the flora that they have in their stomach, so that it maintains and doesn't cause the irritable bowel syndromes that we have here, with our diets mostly shaped based on what we want and not based on what is available, because everything is available to us during any season. It can either be the prices might increase ever so slightly, but we can have it anytime we want it. And we see that there is an observation that the more money you have, the more chances you become obese, because you can afford again these foods that are slightly expensive but very high sugar content and all of that. So when people that are obese, the observation is that because they have many diseases like hypertension, cardiovascular diseases, gallbladder disease, or either respiratory diseases, or even some cancers developing, they are at a higher increase of premature death. And with the fact that in our country we have a high propensity to be more obese, there's a good market now for weight loss. So there are many kinds of diets that exist. We have some diets that they call the Pritkin's diets, where you eat high carbohydrates and high fiber, eating whole grains and vegetables. And then some would say eat high carbohydrates and high protein fat, and the South and the zone diets, low carbohydrate and high protein. So they have different kinds of diets for different things. The goal is how to lose weight. And weight is a function of how much we eat, how much of what we have eaten is not converted into energy because we are not doing enough work to burn it out. So it is stored, the body stores it as fat, thinking that you thought that you were going to need this energy in the future. So we are conflicted between we enjoy our food, it gets fat, but we don't like to exercise, or we don't do exercise enough to be able to burn it up. So our body stores it as fat for the future, which we'll never need. Now, like in any society that is high-tech and solution-driven, we have drugs now that are on the market like Ozempic. It was a drug that was designed to control type 2 diabetes by reducing appetite and slowing the emptying of the stomach, because the more your stomach stays full, the more there is a need for you wanting to eat. And then So with all of these drugs, people say, oh, maybe this is a good deal. The good observation, preliminary observation of the drug is that it is actually making people who are taking the Ozempic thing is making them lose weight. But like any other drug, we don't really know all the long-term effects associated with using the drug to try as a way to lose weight. That is still to be determined several years down the road. Before the Ozempics, other ways in which people were dealing with obese people or obese conditions was gastroplasty, where you can make the stomach smaller by stapling it, because you know the stomach expands, right? It has the folds inside that expand. If you staple some of it and then you eat, you get full fast quickly, because if all these areas that can expand have expanded, your stomach will feel full and then send a message to your brain to say to stop eating. It's a gastric bypass, attaching the lower part of the small intestine to the stomach, kind of making sure that almost like shortening the absorption portion of the length through which the absorption happens. So it makes sure that your stomach is full, but you can only absorb so much food before it goes into your large intestines for ultimate defecation. Gastric banding. Okay, again, reducing stomach size. And then we have liposuction, where they go and put like a probe into the adipose tissue, the fat tissue, and then they kind of warm it a little bit and warm the fat a little and then suck it out. But all of these situations are situations where people go and take the attempts to reduce the amount of fat they have on their body, because there is always a big chance that when you don't lose weight and when you are obese, you could have all of these diseases that I mentioned: diabetes, hypertension, cardiovascular disease, and all of that, and you can lose your life early. So to be able to understand diet, sometimes our nutrition will look at, well, what are the foods that we normally eat, and what does our body need them for? What kind of nutrients do we get out of them, and what do they do for our bodies? I say a nutrient is a component from food that is needed by our body to perform a physiological function, for our body to work. The nutrients that we have include carbohydrates, proteins, lipids, minerals, and vitamins. And then carbohydrates, we have them made out of monosaccharides, and then they would connect to each other to form polysaccharides. They have to be digested. We eat them in this form that they are polysaccharides. They have to be broken down to the simple sugars that we can use to be able to generate energy. As we said, through the process of cellular respiration, they generate energy in the form of ATP that allow us to be able to do work. I said, then the other one source will be like refined grains, because when you have some carbohydrate sources where the fiber on them has been removed, like things like the flour, bleached flour, to make sure that it's white, you remove all the fiber from it. They use it to make bread, cookies, and stuff like that. Those they don't have the vitamins and the fiber. So yes, the only thing that you're gaining from it is the sugar part of it. We need fiber in our diet to be able to make, even though we don't digest it, but it allows our bodies to be able to pull water into our digestive tract to make sure that the food is tracking properly within our digestive system, and then so that we can defecate it easily. Then we have complex carbohydrates. They are complex because they have the vitamins, they have the fiber, and the sources that you can get this from can include whole grains, beans, nuts, and fruits. This is what the kind of carbohydrates that they suggest that we recommend that we should have, because it has the fibers, it has the sugars, yes, but in addition, it has vitamins and minerals present in them. Meanwhile, the ones that are refined is what we do not want to. We want to go easier on them. You can snack on a cookie, a piece of cake, but don't make it like a major piece of your meal, because you're going to be losing a whole lot of other stuff. Can carbohydrates be harmful? I said yes. Refined sugars and fructose sweeteners are not good for us. Because first of all, when you eat a refined sugar or fructose, your body has less work to do in even digesting it. All it has to do is just it's almost like ready for you to absorb so quickly. When you have introduced a large amount of sugars into your bloodstream, it tasks the pancreas to secrete large amounts of insulin to deal with it. And if you do this often, you could have insulin resistance and the type 2 condition, type 2 diabetes condition, that is a challenge because of lifestyle. And then if you have too much of this and the glycogen storage areas in the muscle and in the livers are filled, the next thing is what your body will do next is, I guess we need to store this somewhere else. They convert it into fat and store it. The more fat you have converted, the carbohydrates converted to fat, then we start dealing with this obesity problem that we're talking about. So carbohydrates, eating the wrong type, eating more of it, can cause issues for us. So you would hear to reduce dietary sugar. These are some few things that they suggest that we do. You eat fewer sweets, eat less, drink less, like sodas, ice cream, pastries, eat fresh fruits, not canned fruits. But if you only have options to canned fruits, eat those that are not supplemented with heavy syrup. Use less sugar, less honey. There are some people say, well, I don't use sugar. All the sugar that I use is honey. And yeah, honey still contains sugar. So if you are not taking the sugar that comes like cane sugar or powdered sugar, you think that because of that is great. No, you're not. That's not great. Because you can, if you're using maple syrup, it's sugar. If you're using honey, it's sugar. And then, worst of all, we also have some sweeteners for breakfast, alternatives to sugar, where people would have these alternatives that sometimes our bodies do not even know how to deal with them. As we had fats in the form of hydrogenated fats like margarine, that our body could not deal with it, and then causes more issues than eating regular fat, but in small amounts. We also see that there's also evidence that if you eat less sugar, that is the regular sugar produced from either sugar beets or sugar cane, you are better off than eating some of the sugar substitutes. And then still in reducing dietary sugar, less jam. And then when cooking, instead of using sugars, because most of our foods has sugar added to it, when you include spices such as cinnamon, they will still give you the same kind of flavor and appetite, make the food much appetizing as you would, because most of our foods have sugar there to make us more palatable, make it more palatable for us. This says, do not put sugar in your tea or coffee. That's a tough one for me because I put some sugar in my coffee And I drink coffee black only when I don't want to drink it that fast. But sometimes, but the idea is look at your overall sugar intake if this is a concern for you, and make sure that you do not drink, you do not eat much of it. And again, there is sometimes our ability to test whether something is sweet can be titrated down in such a way that, let's say somebody was making coffee for me. I started, in each time I was having coffee, I was putting 3 cubes of coffee in my cup. Then I would drink it and say, yeah, it's sweet enough for me. If somebody was making my coffee, the next couple of times they put 2.5 cubes of sugar inside, I will still drink it and think that is still fine. Then if they're still titrating it, reducing it to 2 cubes, they can condition my mouth that over time, they reduce the amount of sugar in my coffee to say 1 cube. And because my test buds are conditioned, they could still be sensing, I'll still be having the feeling of it's sweet enough. So you can, I say, avoid processed foods made from refined carbohydrates such as white bread, white rice, pasta, and limit potato intakes. That's just some ways in which you want to reduce the amount of the things that you can eat, and then out of a sudden they produce a surge or a need for insulin, calling the high glycemic index carbohydrates.
Professor?
Yes.
I got a question about pasta because I like eating pasta. What about replacing it with a whole wheat pasta? Does that do you some good as an alternative?
I will ask you the question. What's the difference between whole wheat pasta and regular pasta?
Yeah, sure. Yeah.
So you remember rice, even rice, right? It doesn't come in itself like white rice. When you look at rice, when you take rice, have you seen when they beat them and then the rice grains come out? They're kind of brown with stuff sticking on them, right? And people go at lengths now to clean up all of the brown things, to polish it, to remove all of those things. Those things that you remove from there is where the vitamins are, is where the fibers are. So if you strip the source of these carbohydrates, you clean them up, clean in quotes, you clean them up and remove the brownness that causes the brown rice or the brownness that you use to make brown pasta. If you remove all of those things from there, basically you are stripped, it looks nice and clean, and the off-white color that it looks is thanks to the fact that they have removed all of those other things that are nutritious for you from there. So I would say you are better off eating if the brown pasta that still has some of it. But even when you go to eat the pasta, so you have a choice: brown pasta or the other kind of pasta. Go with brown pasta. Question next: if you are going with brown pasta, what does that do for you? It is helping you out because it has some fiber in it. It has some vitamins still retained in it and things like that. Now, the question is, should you eat fiber every day? Then it becomes a different question. Do you add some sugars when you eat in your pasta that the way you make it? You do, I don't know. So the whole idea is, if you are as we go through this, we'll go, okay, well, what would be the recommendation of how we can eat? So to your point, I would say, given choices of pasta, I will go with the brown one. Did I answer your question?
Yeah, thank you.
Okay. Now, it's about 1:21. I want us to end here so that it is raining outside, because I want us to have enough time to get to the lab safely. We can end here, it's okay. And we will continue pick up from here next week. Does anybody have any objection to that, or anybody has any questions before we leave?
The slides you showed before this one, like before our break, are those slides already available on Canvas?
Can you check and let's see? I think it should have.
Because I tried looking up for them because I was during my break I checked the study guide and it was talking about where was it?
So you're talking about the slide.
Because you were mentioning in the previous lecture about what pregnant women worry about, the RF. I don't know where that slide is on the
Oh,
canvas.
Yeah, the blood slides.
Okay,
Oh,
let's
it's on
not
those
there.
ones?
Yeah,
yeah.
it's not on the modules. So will that be available on Canvas later?
I thought it should be. Can you check right now? Okay. Let me share my screen with you. Okay. Let me share my screen and look at the cardiovascular system. Oh, okay. Okay, yeah. Are you sure you're not seeing them yet? I can see why now. Thank you for pointing that out. I don't know. I published everything on there. They didn't publish. Check. Can you check now and see whether you can see them now?
Yes, it's there now.
Is anybody checking?
Yes,
Yeah,
I
it's
see
there
now.
now.
Perfect. Thanks for pointing this out. Let's, yeah, if you need just look out for stuff again. If something not sure like this, I'm glad you brought this up. So let's catch up in the lab later, okay? All right. I'm going to close the lab for today, the lecture for today. I'll see you in the lab.
Thank you. See you then.
Thank you.
All right, see you.